All right. Well, hi everyone. Happy Friday, and welcome to Translate, a webinar on translational research in radiology. We have with us today Dr. Jacob Hooker and Dr. Joshua Roffman. Dr. Hooker is a professor of radiology at Harvard Medical School, director of radiochemistry at the Martinos Center for Biomedical Imaging, and now also scientific director at the Lurie Center for Autism. In his work, he has sought to accelerate the study of the living human brain and nervous system through the development and application of molecular imaging agents. His lab has introduced several technologies for neuroscience, including a first-in-class radio tracer for neuroepigenetic imaging. Dr. Roffman is a board-certified psychiatrist and neuroscientist who directs the Early Brain Development Initiative at Mass General. He has also served as co-director of Mass General Neuroscience and as director of research for the MGH schizophrenia clinical and research program. The goals of his research program are to discover, develop, and implement early life interventions that protect against risk of neuropsychiatric illness in young people. He is an associate professor of psychiatry at Harvard Medical School. Just a quick note before we dive in. If anyone has any questions at any point, please feel free to drop them into the Q&A, and we'll address them either during the discussion or at the end during the Q&A session. So, given that, if I can start with you, Josh, if you could describe some of the clinical problems in psychiatry that imaging could help address, and what progress has been made thus far in doing so.
Thanks, Gary. Thanks for the invitation to be here. It's always great fun to talk with Jacob about science, and thanks to everyone who is out in the audience. Really looking forward to hearing your thoughts as well. So. As a clinician who has kind of grown up around in my career, recognizing that the treatments that we have either to treat or prevent numerous psychiatric illnesses are woefully lacking in comparison to other areas of medicine. I and my colleagues have looked to the brain for answers, since we know that this is the seat of these disorders, unless we're missing something really bad, but no, we're pretty sure it's not the liver. Imaging, of course, provides a whole array of tools now for interrogating brain structure and function, and hopefully leading us to individual type differences that are clinically useful, again, either in service of diagnosis, prognosis, treatment, prevention. We think about how echocardiograms are used in cardiology, and think, well, can imaging of the brain provide us similar handles, similar opportunities? As someone who's been at this now for more than 20 years after finishing medical school and then through residency training, and looking at on one hand, the really spectacular technical advancements in imaging and related technologies since then, and at the same time, unfortunately, the lack of significant development on the side of interventions in psychiatry and in service of better outcomes. One can't help but notice that disconnect, that imaging has not yet achieved impact on the level of improved clinical outcomes. And the question is, can it? And as someone, again, who's been in this area for a long time, I continue to be optimistic that it can. And at the same time, we have to be humble and pay attention to the fact that so far it really hasn't. There's no psychiatric illness where imaging of an individual patient can help guide diagnosis or management, or even risk stratification. We're learning a ton from research studies involving patients about circuits that are involved in these illnesses, about the time course through which alterations in those circuits might take someone from the state of being predisposed to an illness to the point where they actually develop an illness. And there are some fantastic ongoing longitudinal studies in the US, Europe, and elsewhere that I think will be of tremendous service by starting early in life and following individuals with serial scans through these windows of increased risk. I think we're going to learn a lot, and I hope that we will learn something that is actionable. But at the same time, the reality where we are right now is that imaging has not translated to improved outcomes, in psychiatry, as it has arguably in other areas of medicine. And that's where it's so important to work with people like Jacob, who are on the cutting edge and developing new tools and new technologies that give us much more resolution, not just in terms of physical, spatial temporal resolution, but on the level of brain biology that ultimately we're going to need to be able to move the dial on this. So Jacob, why don't I turn it over to you and hear your thoughts on imaging and psychiatry and whether we can bridge this divide.
Yeah, thanks for that overview, Josh. When I think about imaging and where it has impact, you can typically draw a corollary to pathology, right? Look at neurology as an example, another adjacent field where you say imaging has had impact, right? We have diagnostic scans for many neurological diseases. And so, why hasn't that translated, right, in psychiatry? And I think it really comes down to the fundamental lack of understanding of etiology, right? In neurology, where at least there's a pathology hallmark or some basis for understanding what's going wrong, we seem to be able to translate. We can figure out how to make imaging technologies that give us clues. In psychiatry, imaging has been successful in elucidating a lot of basic science mechanism, right? Narrowing the whole brain down to individual circuits or brain regions that may be most likely to contribute to disease. And I think imaging has been able to confirm a lot of, scientific anecdotes that connect neural circuitry to disease. And so I think we're at the cusp of that sort of revolution in psychiatry, where we start to appreciate through whether it's genetic association, risk variation, genes as they lead to phenotypes, pathways, how to design imaging technology that will ultimately help in ways that it has in neurological science generally. And one of the things that Josh and I often talk about is this disconnect in psychiatry between genetic risk and how that really plays out at the individual level. You could be at high risk from a genetic association perspective, a sort of polygenic risk score, if you will, for mental illness, and yet you're way more likely not to develop it than to develop it. Even the highest risks associated with genes and polygenic risk in psychiatry really only increase the probability, by 70 or 80%. And yet, given the rarity generally, if there's a 1% prevalence, what that means is now you have instead of a 1.1% chance, you've got a 1.7% chance. So how do we move from polygenic risk to this notion that other features are at play that we have to measure? And that's where, Josh and I have always sort of aligned on this notion that epigenetics, or sort of the interplay between that genetic risk and the environment, might be mechanistically a way to sort of connect the dots between what we know as abnormal biology or could be abnormal biology and how it manifests over one's life. Of course, there are a lot of challenges to that that we can talk in part through here. But I think the opportunity is pretty profound, because environmental factors, life events, can be quite, not deterministic, can be quite predictive of sort of major increased risk. And so, we have set out to work together to sort of operate in that space, develop tools and technologies, concepts translationally that would allow us to sort of look at the environmental impact on your brain's ability to function. And so I'll let Josh describe some of his work in understanding the connection between, say, DNA methylation, folate and methylfolate, and some of the some of the concepts that then lead us to get really excited about imaging technology that can help map on what may be going wrong there.
You'd think by now I would know where the unmute button is. Sorry. Yeah, so that's one way in which I think imaging can really be exciting. So start with a population level finding. Even, you know, natural experiments around famine studies from World War II and in China in the late 50s and early 60s. And there are these really pronounced relationships where people who were born during those times have you know, substantially higher risk of developing serious mental illness like schizophrenia. Clearly this is the environment at play in the developing brain, even in utero, with a really substantial effect size, and yet the number of intermediate possibilities there in terms of different levels of biology, specific versus general exposures, is mind-boggling, right? We talk about the genome in terms of, you know, sequencing and genome-wide association studies. Increasingly, there is talk about the environment in the same way, and trying to figure out which pieces from the environment are mediating these effects is an important challenge, again, simply because of the effect size. So something like folic acid is an interesting candidate. You know, again, this starts from epidemiology in those people who were born during famine who go on to develop schizophrenia. There was a disproportionate number who also had spina bifida at birth, which is a neural tube defect, which we know after decades of research in the UK and other places tends to be exquisitely sensitive to maternal folic acid. So when mom gets folic acid on board early in pregnancy, the risk of neural tube defects is substantially diminished. So, you know, might it not just be neural tube defects? Could folate be doing something else in concert with other environmental factors? So we've started to look at that in adolescence, having, you know, spent some time in the clinical trial space in chronic schizophrenia, looking at folate-based interventions. And we're seeing some interesting patterns where kids who were exposed to higher levels of folate early in gestation tend to show different patterns of brain development than kids who were not. And I think even more importantly, and this is not our work, this is other, you know, large public health studies, there is a pretty robust relationship to autism risk, where moms who take prenatal folic acid starting before conception, or at least start with folic acid on board within the first couple of months of pregnancy, which of course is after the neural tube closes, but still may be relevant in terms of early brain development. Those kids, compared to when moms don't start folic acid until later in pregnancy, have roughly half the risk of autism. Now, I don't know if I believe that amount of reduction, given no, these are big, well-controlled studies, and still, I don't think folic acid is a silver bullet, but it's proof of concept. That early environment can be modified in positive and protective ways, and not just, you know, detrimental ways, to influence brain development throughout the lifespan. So the question for folate is, great, we can, you know, put these kids in the scanner and we can look at brain structure and brain function. But again, how far away is that from what folic acid is doing on the level of cell and molecular biology? Like, for example, methylation. That's what folic acid does basically in the body, is it provides one carbon moieties for transmethylation reactions. How do we measure that? We can look in the periphery, we can look at peripheral methylation, but the problem there, of course, is that peripheral patterns of methylation may not, and probably for the most part don't, reflect what's going on in the brain, or even in, you know, specialized tissue within the brain. So what we really need is a way to look within the brain itself to understand, you know, not even folate effects, but more broadly, what's happening on the level of epigenetics in the brain. And that's where PET and Jacob's work and colleagues with Martinos comes right into play. So Jacob, do you want to describe a little bit of how you got to Martinos and what it's telling us about differential methylation and potentially down the line gene expression in patient populations?
Yeah. So more than a decade ago, I got really fascinated by this notion that we might be able to image environmental impact in the brain. And in thinking about all of the different epigenetic mechanisms, remember, epigenetic mechanisms are profound, right? They define very simple marks. I'm a chemist, and very simple changes, a methyl group, an acetyl group, these little tiny changes in one or two positions of your genome or on chromatin, the balls that your DNA are sort of entwined in. Really small, subtle chemical modifications can determine the cell fate. They can reprogram a cell from being a skin cell to a heart cell, right? So when you imagine dysfunction at the epigenetic level, it's not like some small issue. It can lead to hundreds, if not thousands, of genes downstream being dysregulated. And we thought, well, if we could image just the general health of the epigenetic processes that are going on, maybe we could start to link those up and say, well, the enzymes, the machines that put on these marks look healthy or not, and then we can sort of map the consequence of that. So it was a pretty naive idea. But I thought if we could do that, it could become a platform where we start interrogating all the different sort of machines that operate at this epigenetic level. And maybe we could stumble upon, through a little bit of luck, some space that would help us understand how to do this with time. And so we started looking at all the different space around epigenetic modulators, how to create imaging agents out of them, and really took a look at cancer biology where epigenetic manipulation was being used therapeutically. And so there were drug-like molecules that engaged these machines and that help reprogram, change what cancer was doing in ways that chemotherapeutics would be more efficacious. And the one protein target that we became fascinated around was actually a whole class called histone deacetylases. But in the brain, very specifically, there's a subset of those enzymes, that what I'll call HDACs, that really define memory function, that are really important in neurodevelopment, and that had been associated to some degree with neurodegeneration. So given that we had a sort of cancer playbook for how to develop these molecules and a really important connection to neuroscience, we started there. And through a tremendous effort of a huge number of people that I've worked with over time, we developed a small molecule that engages those proteins, the class 1 HDACs. And we figured out how to get it into the brain with an imaging label on it, an isotope that allows us to see where that molecule goes through positron emission tomography, so that it could report on what was going on with HDAC. And so keep in mind that PET really works like I label this molecule, but I don't really care about the molecule. I get it to the brain, it engages with what I care about, HDAC, and I can now see that, sort of a surrogate representation of that enzyme. It took us, I don't know, 7 or 8 years to develop the molecule and get it to people. And we've spent the last, I don't know, five or so years exploring the use of that technology in neurological, psychiatric, neurodevelopmental disorders, and more. And I think what's been satisfying to see is that we can detect differences. So epigenetic dysfunction, if you will, aberrant regulation of these proteins. There's something different about them that tends to be specific to disease, right? The regional signature in the brain, where things are different in the brain, seem to map on to some other observations of circuit dysfunction. And so I think we're now at that cusp of now trying to understand, well, when in the course of disease does that occur? Is this cause, consequence, or neither? And how does it manifest change? And most importantly, could those signatures be used in therapeutic concepts for those people that are most dysregulated in those ways? Can we develop drug intervention strategies that would rescue that behavior? And so, Josh has seen some of the work sort of in particular in bipolar disorder and schizophrenia that helps sort of connect to some of the neuroimaging work that he's done, for example, in schizophrenia and risk around that I'll let him describe.
Yeah. So and that last piece, I think, Jacob, is the theme, right? It's not imaging for imaging sake, which isn't to say that imaging for imaging's sake is what motivates people to do this work. It's not just case control differences, which we've been able to demonstrate across a whole slew of modalities and across a whole slew of neuropsychiatric disorders, even if at not on the individual level, at least on the group level. It's how do we modify the system, and can we measure how we're modifying the system? So this is a molecular entree, basically, it's a platform. You have a new intervention that you think may be altering the epigenetic milieu in the brain. And maybe even better, you have someone, as Jacob alluded to, who seems to have the substrate wherein that kind of modification could be more meaningful because of where someone is starting on the curve. Now, all that being said, I think both of us would agree it's unlikely that neuroimaging, PET neuroimaging, in particular, given the cost and the logistical issues, will ever really be part of mainstream diagnostic therapeutic protocol for psychiatric disorders. But it needn't be in order to be clinically useful. So, one example that I really like from another area of medicine is in cardiology. We all know now that statin medications, which help control cholesterol and save probably hundreds of thousands of lives every year. How were those drugs developed? Well, it started with a rare, highly penetrant mutation in the enzyme HMG-CoA reductase, which plays a pivotal role in metabolizing cholesterol. Does that mean that we have to sequence the whole population before seeing if they're going to benefit from a statin? No, it was focusing, intensely on that group that led to the development of drugs that target that enzyme, and that's where the statins come in. And the ironic thing is, if you look at genome-wide association studies in cardiology, to this day, HMG-CoA reductase, which is the enzyme for one of the most important treatments to have come out of cardiology research in the last 50 years, HMG-CoA reductase itself is not a GWAS hit. And yet it was used to develop a therapeutic, which is incredibly meaningful. So by way of analogy, I think that's our hope for what these imaging tools will enable us to do, is understand biology in an intensive way that then lets us think about new intervention development that will go beyond what we are able to see just with imaging.
These psychiatric disorders are incredibly heterogeneous, and one of the challenges that we face is how to homogenize, like how do you get down to like a subset of what we call schizophrenia that might look similar enough where we could get to that equivalent that you just referenced in cardiology. So Josh, when you think about patient stratification, sort of clinical features, different concepts for how we cluster so that we can maximize what our imaging tools tell us, right? Because our imaging will be fundamentally limited by how we make choices in who we're measuring. How do you think that part of psychiatry is evolving in ways that may allow us to take advantage of it?
Yeah, great question. So going back to GWAS, All of these, ginormous psychiatric genomics consortium studies, which now have hundreds of thousands of participants, are effectively case control studies, right? You have your schizophrenia pool and you have your healthy pool. You have your bipolar pool and your healthy pool, your depression pool and your healthy pool. And one of the things that we've learned, which is not terribly surprising as a clinician, is that the variants, the genetic variants that fall out of these analyses actually tend to overlap pretty substantially. So even though we're looking at somebody who has schizophrenia, who clinically looks very different from someone who has depression or bipolar disorder, it turns out that some of the genetic liability overlaps. And in tandem, we're kind of beginning to rethink, at our peril, because, in psychiatry, there have been pendulum swings for, the last 100 years in terms of how we think about psychiatric nosology and lumping versus splitting. But I'll tell you that the current thinking, it seems to be better aligned with that underlying biology. And remember, most of these disorders are strongly heritable genes. Is a very good place to start. Starting to think about not schizophrenia, but psychosis. Not just depression, but rumination or anhedonia, which is loss of pleasure seeking. These syndromes really stretch across DSM diagnoses, and the whole impetus behind this National Institute of Mental Health RDoC initiative, which you may have heard of, is that there may be a tighter relationship between cellular and molecular underpinnings of disease and clinical syndromes that are continuous and even continuous out to the level of the general population, rather than focusing on this dichotomy between disease and non-disease. And again, this is where imaging potentially adds some heft, because in imaging, we're not looking, in neurology, someone's either having a stroke or they're not. In psychiatry, there's going to be a continuous, Gaussian distribution of your imaging phenotype of interest. And understanding disease on a continuum, it really opens up some degrees of freedom in terms of exploring the underlying biology and its, related continuums. So I think, breaking that box of case control is potentially really important. And, it not only is it refining how we're doing imaging studies, it's, beginning to refine how we do genetic studies, not just being satisfied with schizophrenia versus healthy, but evaluating people using continuous measures of psychopathology and seeing how those continuous measures relate to underlying genetic architecture. I think that's the next big advance in terms of how we think about these large-scale genetic studies, and certainly in imaging, we've been doing that for a while. And is it surprising then that with Martinos there are differences in schizophrenia and in bipolar disorder, and potentially autism, which is something else that I know Jacob and the team are looking at? No, not surprising, given the importance of epigenetics to each of these syndromes. But we may be beginning to see how there are differences in the brain across those syndromes in terms of relationships between the imaging markers and clinical presentation.
One of the challenges that we face in neurodevelopment and psychiatry is, of course, time, right? That a lot of these processes have set course in utero, maybe in early childhood, adolescence. There's risk in teenage years. There's just a tremendous sort of spectrum of when insults can occur on the time course that sort of define ultimately what we see later as manifest disease in your 30s or 40s. And so, I think we have this added dimension that has made imaging in psychiatry a bit harder to sort of crack, in that we tend to have to work backwards a little bit from manifest disease back to some prodromal state, back to some, causal biology. And that's really tough. And so, one of the things we're trying to do is start in, adults where we can characterize disease, whether it's along an axis of or some symptom axis or some other, and start correlating imaging with something we can measure, say peripherally or otherwise, that allows us to go earlier and earlier in time. I am encouraged that there are also efforts, though, at the Martinos Center, for example, to build next generation scanners that may enable us to use, say, positron emission tomography, which uses radioactivity and generally is not used in children. But these new technologies may allow us to move in that direction, that as we get higher sensitivity scanners, as we reduce the amount of radioactive dose that we need to give, I think, if I were to make a prediction over the next 10 years where we'll see this go, is that technologies that now are limited to adults really become research tools that we can apply in children. That said, I don't think we'll ever get around this notion of connecting the science through peripheral marks. And so Josh, I know you think a lot about this because you have the same challenge in time, right? How do you connect the dots along the time course? Given that the periphery is not the brain, right? Like, things that we measure peripherally may or may not relate. How do you sort of find validity when you see a peripheral observation and try and connect that to the brain?
Yeah, great question. So I'm teaching second year medical students now at the HST program, and we're talking about phenomenology, and we're talking about, for major depressive disorder, you need to have, either depressed mood or anhedonia in combination with 4 other symptoms, and there's a list of 8. So there are thousands of potential combinations. And those combinations overlap with other psychiatric disorders. So how do you get to a diagnosis? Well, you get to a diagnosis through triangulation. You say you've got this and this and this and this. And with each piece of information, the chance of getting to the right diagnosis gets better and better and better. So I tend to think about peripheral markers in the same way. I think they can be useful. The question is, are they useful, and are they useful in a way that is convergent with what we know is going on in the brain? And it's a challenge to get that in vivo in humans, although increasingly people are coming back to an old target in CNS research, which is CSF, right? If you can see what's going on behind, at least going behind the blood-brain barrier, perhaps you have a better chance of relevance. But it takes that. It takes postmortem studies. It takes animal models. It takes human-induced pluripotent stem cell studies. You have to have multiple lines of evidence that triangulate. And again, if the upshot is that there's a peripheral measure that has some sensitivity and is non-invasive, then absolutely, that's useful information.
Yeah, I mean, we've seen tremendous success there. If you think in neurodegeneration, where Alzheimer's disease was characterized clinically, right, or at least diagnosed as a probability, until we had the ability to look in the brain and confirm amyloid plaques were present, right? And so the consequence to that early ambiguity in diagnosis was that even in early clinical trials, a huge proportion of folks that were enrolled in clinical trials with the diagnosis of Alzheimer's disease didn't have it. And so, it's hard to homogenize for a clinical trial outcome if you don't even have the tools. And there was a lot of skepticism, in the early days of PET imaging as it related to the diagnosis of Alzheimer's disease, and there still is because of access. And yet that imaging tool fundamentally enabled us to know, to confirm diagnosis that allowed us to take CSF and then blood and serum samples, right? To finally say, well, can we find an amyloid ratio or some precursor or some marker that would strongly enough correlate with the imaging verified pathology that we could diagnose through, say, CSF, lumbar puncture, or through now blood, right? And so in my mind, particularly in neuroscience imaging, and I see this where psychiatry goes, as we start to find these, what I'll call anchor biochemical findings in the brain through imaging, right? Imaging and sort of hard basic science from genetics forward, we will then start to connect the dots between that to CSF and then to blood. And eventually we will be able to triangulate, like Josh, I think you're mentioning there, where, maybe we will have objective ways to quantify at least a neurochemical signature that we call, whether we call it schizophrenia or something new at that point, we'll be able to classify a disease differently. And so I do think imaging is often maybe a first step in getting to that validation of the secondary deployable technologies.
Absolutely. And, we would like to open this up, by the way, to conversation. If you have questions or comments or want to contribute something, please, I guess you can either raise your hand, we can turn on your audio, or you could just stick it in the Q&A. While we're waiting, maybe for some of those questions to formulate, just to put in a plug, what Jacob was just mentioning in terms of the ability to gather together all of these marker types from the same individual, be it from imaging, peripheral markers, CSF markers, and further, to do so in the context of a clinical trial, is now something that is really, enabled at MGH and Martinos through the Interdisciplinary Brain Center, which is just opening up its facilities on the second floor of Building 149 under the direction of Steve Arnold. It's a tremendous resource for translational, clinical translational research, and again, the ability to really get rich multimodal data from the same individual over time is really remarkable. And so if anybody has any questions about that, feel free to reach out to us or to Stephen Arnold directly, who I'm sure would be very happy to hear from you. Let's see, any questions?
Actually, while we're waiting for questions to roll in, maybe if I could ask a quick one. So, the central question of this seminar series is how to advance the translation of biomedical imaging research into clinical practice. And when we were talking the other day, Jacob used a phrase I really like. He said, historically, technology developers have developed technologies and then thrown them over the fence. And then similarly, physicians have, teased out problems that need to be addressed and then thrown those over the fence. But we're seeing in recent years that, we're seeing a blurring of these lines. So I wonder if you could comment on, sort of how this has played out and what it could mean for psychiatric care.
I mean, I see a tremendous blurring of classic labels of clinician, researcher, and so forth. I know undergraduates often agonize over, do I go to graduate school or do I go to get my, MD? Do I do an MD, PhD to enable research? And I think I think you can approach in the current way we're trained, you can approach the problem from either direction. Being classically trained as a physician like Josh is a medical doctor, MD, card carrying psychiatrist, and yet does very fundamental basic science, right? Fundamental mechanism, works with bench science, all the way through models forward. On the opposite end, I was trained as a very fundamental basic scientist. And yet now a large part of the imaging program and work that I do is focused on patients and patient observation. And I think that blurring has become really important in thinking about how technology and unmet needs are matched more efficiently, so that we can understand the context of use when developing technology, so that we don't develop technology that has no context for use ultimately. Josh, your thoughts on that?
Yeah, no, I think the environment is finally, the milieu is finally coming to realize that none of us can do this on our own, right? We, clinicians don't have the answers by themselves to figure out how to address, these vexing problems of treatment and prevention. And people on the basic science side have great tools and great science and great technology, but are the questions that they're addressing ultimately the ones that are going to be the highest impact on the level of public health? And I'll tell a quick story. I mean, first of all, I should say the only thing worse than what I'm about to describe is when there is clinical research that does not know of or fails to take advantage of the opportunities to connect to translational and basic science in a way that could support what they're doing clinically. Sometimes it goes the other way as well. I remember reading a paper a few years ago in Science or Nature, I can't remember which, and it was about one of the receptors that is targeted by one of our treatments in psychiatry, and it was a beautiful study. And elegant science. And the problem is that the frame of the article was, now that we understand this receptor, we're really cooking. Now we can really get to some better treatments for psychiatric illness. And the problem is actually that receptor is now more implicated in medication-induced side effects than it is in the efficacy of the medication itself. That's a disconnect that just shouldn't be happening. And that's why I feel fortunate to be here, right, at MGH, where we have not only multiple layers of expertise across different backgrounds, but we actually talk to each other. A place like Martinos, we're actually working next to each other, which is fantastic when we're actually, there. And these days it's been a challenge, but I can't remember the last time I saw Jacob in person, but it's been a while. But even something like Mass General Neuroscience, which is designed to try and articulate these synergies in service of better clinical outcomes. That's why I think we're all working in the right place. And if it's going to happen anywhere, I think it's got a good chance of happening here.
Josh, I just saw one come in that I'll read to you because I think you'll probably have great insights into this. I was wondering what has been done in finding correlations between neurodynamics, for example, EEG, MEG, resting state, functional MRI, and psychiatric disorders.
Sure, great question, and quite a bit. Across all the modalities that you mentioned, in recent years, probably resting state fMRI has gotten the most attention. And indeed, there are certain patterns that are characteristic of certain disorders. Whether they're specific to certain disorders, I think remains an open question for the reasons that we were talking about before. So the challenge, though, and to kind of reflect on the theme of this, is not just to show that there is a relationship between a brain signal and a certain disorder, a correlation, as you said in the question, but is that meaningful? Is it tractable? When I think of the unfortunate fallacies in psychiatric neuroscience is, if you can measure something in the brain, it must be important to disease or to treatment. And, some of the time it is, and some of the time it isn't. So what gets us beyond that is experimental designs with manipulation of those signals, be it through a treatment or direct stimulation, like with TMS, if you want to interrogate a circuit, and then seeing how those changes are relevant to clinical outcomes. I think that's even richer than correlational studies, and I really think that's really the future of psychiatric neuroimaging. Between that and shifting from kind of group analysis levels where you can't really see individual differences to things like what Randy Buckner and others are working on, where you're doing deep, intensive, repeated measures within the same individual over time, and you can elaborate those networks and manipulations in those networks on the level of the individual. They're doing it in TMS as well in terms of individual targeting. That level of individual variation and really, instead of it being something that gets in the way by creating noise, rather leveraging those individual differences to develop personalized treatments, that I think at the end of the day is what a lot of people are excited about.
Yeah, I agree that one big area of opportunity moving forward will be this massive growth of interventional psychiatry of all sorts, You're seeing all sorts of new hypotheses on neuroplasticity in psychiatry. How do psychedelics, for example, manipulate and remodel brain activity? Fortunately, the imaging is ready for that field to emerge, or fields like that, TMS, deep brain stimulation. Those techniques, when the imaging batteries that we have, if you will, combined with these new modalities for therapy, I think will sort of teach us a lot about what we can and can't do in terms of manipulating brain function, particularly in adults, once things have sort of gone awry. So I'm really excited about the connection between pharmacology and understanding if imaging can predict better what kinds of drugs work for who, whether we can fundamentally see remodeling circuitry through neuromodulation. I think it's a really exciting time to see these things come together.
So there's a question from Maria about machine learning. Will it play a role in translating brain imaging to clinic? Jacob, do you want to take a first stab at that one, or I'm also happy to?
Yeah, I think to some degree it will, and I think much like in other aspects of imaging, we'll start to see the early fruits of that in other space, probably not psychiatric illness. We're more likely to see machine learning and other AI kind of based techniques help in cardiology and understanding, lung function, maybe neurology. Maybe there's some signatures in sort of high content or high resolution MRI that we'll be able to pick out that we couldn't have seen without those kind of techniques. I think it'll take a little while longer because of the heterogeneity in psychiatry, because it's such a multivariate problem. Machine learning takes a huge data set, and then when you have a heterogeneous population, you need that, to the nth power on every dimension that the disease is different. So I think it's got a ways to go, but we'll see early fruits of machine learning and imaging quite soon, and its clinical use. And then I think psychiatry will come in time.
Yeah, I think it's a much better answer than I would have been able to give. So I'm glad you spoke up on that first. Question from Amy Peters. Thinking about the discussed gap between imaging and actually moving the dial, new treatments after decades of imaging research. Curious of your thoughts on how do we know when a neuroimaging finding is ready for clinical evaluation? A lot of existing treatments have been either discovered accidentally or tested in research based on biological hypotheses where there's imaging evidence for that or not. So what do you view as reasons not to just continue going after a new target via clinical evaluation? Or at what stage in the imaging pipeline do you think that, do you think there's measurable evidence that it likely yields better outcomes than a traditional clinical trial? Great question. Remember, it doesn't need to rely on imaging, right? If it's effective, and there's proof of efficacy. One thing that I get a little hung up on, I'm going to vent a little bit here, sorry, is, some of the NIH infrastructure for treatment development, especially in psychiatry, relies on basically proof of target engagement, where, you have to add in either an imaging study or a CSF study to show that this is doing something meaningful in the brain. We knew that lithium was an effective treatment for bipolar disorder long before neuroimaging was invented. And we still don't quite know how it works, but we know it works. So is imaging a necessary intermediary? No, I don't think it is. On the other hand, if imaging can be helpful in a clinical trial process, in phase 1, phase 2, to help to amplify the potential therapeutic effects of an agent, and to study that in a small number of people before you go out and have your ginormous clinical trial with 1,000 or 10,000 people, absolutely, that is well worth it. And if imaging provides some biological insights that can be leveraged not only to figure out who's going to respond to what treatment, but frankly, can be used to develop other treatments as new pathways of interest are identified. That's great as well. But I guess the bottom line is, I don't think that imaging needs to be a part of treatment development, nor should it be in many cases, but it can be, depending on the individual situation. Jacob, I don't know if you have any other thoughts about that.
I mean, I generally agree. And I tend to, when thinking therapy, as a chemist, be pharmacologically biased. So I'll acknowledge that. I do believe validating a target through an imaging signature is unlikely in the near term, because we barely know what the biology tells us, much less what the imaging signals do. What I do tend to tell people is that I do believe that for a drug to work in the brain, it must get there and it must engage a target. It's hopefully relevant target, and then relevance will be determined through efficacy trials later, right? So I do think imaging has played a really important role in psychiatry, especially in the early days of dopaminergic, antipsychotic development, understanding dose occupancy relationship, particularly when overshooting on dose can have negative consequences. It's really important to map how much is getting to the brain, how much of the target's engaged, and then how does that correlate with efficacy. So I think imaging plays a really important role in therapeutic development, but not, Amy, to your question on target validation as much per se, at least at this point.
Excellent. Well, thank you so much. And thank you everyone for your questions and for joining us. And thank you, Jacob and Josh, for a really amazing discussion today.
Thank you. And if other questions or thoughts come up, please don't hesitate to reach out to us individually. We'd love to continue the conversation.
Awesome. Well, thank you so much. And everyone enjoy your Friday.
Thanks, everyone. Take care.